Hello, I'm Dena Ladd, the Executive Director for the Missouri Cures Education Foundation, whose mission is to protect and promote medical research in the state of Missouri. Thank you for joining us for our Lunch with a Researcher series. Today's discussion is focused on diabetes research, treatments, education, and community engagement. I am so excited to have you all with us today. We have people from around the state and around the country joining us. During the discussion, please put your questions in the chat box, and we hope to get to as many questions as we possibly can. I would also encourage you to go to our website, MissouriCures.org, to find out more about our 2021 events. Let's get this discussion started. I'd like to introduce our speakers for today. Dr. Jeff Millman, he's the Assistant Professor of Medicine and Biomedical Engineering, Washington University School of Medicine. Dr. Millman came from Harvard, and we kind of stole him away, and now he's with us here at Washington University. Dr. Leslie Hinyard, she's the Director, Advanced Health Data, Ahead Institute, St. Louis University School of Medicine. Derek Rapp, Managing Director, RiverVest Venture Partners, former President and Chief Executive Officer of JDRF, and Board Member of Research America. Welcome, everyone. I am so excited to have you with us today. I know how busy you all are, so thank you so much for taking the time to be with us.
Thank you, Dena.
Yeah, thanks for having us.
Absolutely. So Jeff, let's start out with some basic information. Can you first explain the basic difference between type 1 and type 2 diabetes?
Yeah, certainly. So both type 1 and type 2 diabetes are the end result of two different processes that kind of manifest as the same thing at the end. So in type 1 diabetes, the cells in the pancreas called the beta cell that are responsible for measuring or sensing how much sugar is present in the blood and secreting insulin in response to that in order to manage blood sugar levels. Those cells have been destroyed due to an autoimmune attack. The immune system makes a mistake, thinks that the cells are foreign, and will actually go and destroy these cells, meaning that in patients with type 1 diabetes, they aren't producing enough insulin to control their own blood sugar levels. Type 2 diabetes is different in that there's a few different ways that this can manifest, but typically in type 2 diabetes, these patients have become resistant to the insulin. So it's not that they aren't producing insulin anymore, it's that their body requires more insulin than their beta cells are able to produce. But in the end, no matter if it's type 1 diabetes or type 2 diabetes, there is not enough insulin there to control the patient's blood sugar levels. And so they have high blood glucose levels as a result. And this leads to many complications that can really reduce quality of life for patients, making it a very important diseases that we should be focusing our attention to.
So Leslie and Derek, is there anything you want to add to kind of the basic diabetes 101?
Leslie?
I think the only thing I would add to that really lovely discussion or description is that when we talk about prevention and prevention measures that go in towards diabetes, a lot of times the prevention measures we're talking about really are geared towards the risk factors for development of type 2 diabetes, because type 1 diabetes is an autoimmune issue that we don't really know how to prevent. So that's the only kind of difference in there. Well, we'll get there. Maybe someday we will, and then we'll solve the problem, right? And so from the perspective of this population level that I refer to a lot, we really kind of make that distinction specifically so that we can talk about what measures we're in or what interventions we're using with which types of people, depending on what their need is.
And sorry to interrupt, but yes, so in a few minutes, we'll talk about some of those prevention measures for type 1 also, but Leslie's exactly right. And I think a point I'll just make is that when people hear that word autoimmune, there are many autoimmune diseases. And in fact, there's a high correlation among a number of autoimmune diseases. So I'm talking about diseases like celiac disease and Crohn's and ulcerative colitis and vitiligo and rheumatoid arthritis and psoriatic arthritis and psoriasis and others. And in fact, within families, if they have one of those diseases, they have many. Our particular family has just about every one I just named. We have 7 different autoimmune diseases in our family, including our son Turner has type 1 diabetes.
Well,
And really
thank
trying
you.
to understand the differences in what causes type 1 and type 2 diabetes is very complicated and something that we need to keep on focusing on in order to not just treat diabetes or cure diabetes after it occurs, but the goal here for both type 1 and type 2 diabetes would be if we understand what causes it and the differences that exist there as well. We can just prevent diabetes from ever forming in the first place. That would really be the holy grail, if you will, of being able to deal with diabetes in our population.
And also for you all, the scientists, not I, but it's much easier to stop a metabolic process from ever occurring than to reversing one that's already occurring in the body. So that's one more reason why prevention is so great. So not only do you avoid all the challenges that come with a person first becoming sick, but you make it a lot easier for that never to take place at all.
Yeah. So Jeff, when I went to your lab page at Washington University School of Medicine, it states that you focus on the generation insulin producing pancreatic B cells for the study and treatment of diabetes. And your expertise is specializing in the application of a wide range of state-of-the-art technologies to discover a cure for diabetes. So could you tell us a little bit about your cutting-edge research, please?
Yeah, I'm very happy to do that. So the kind of the backbone of my lab's research is that we have developed technologies and approaches for taking stem cells, which are unspecialized cells that basically need to be taught what to do, be told to become something that is specialized. And we give them the appropriate signals to teach these cells to transform to become a specialized cell type that actually has a function in the body. In this case, in particular, we focus on making the insulin-producing beta cell, which again is kind of the crux of all forms of diabetes and the target of destruction by the immune system in type 1 diabetes. And so this is a very complicated process. It takes us about a month and a half of working in the lab to go from a frozen vial of stem cells all the way to a population of insulin secreting beta cells that we can actually use for study. There's a lot of different technologies that are involved in it. It's not just a matter of being really good at biology to do it. You have to, really understand the bioengineering aspect. It is a manufacturing process as well. So we have my lab consists of people with various different specialties in biology, which are necessary to understand that, but also people with bioengineering backgrounds. And my own background in terms of my degrees are in engineering before I started focusing more heavily on stem cells. I see there's a question in the chat asking what kind of stem cells. So these are what are referred to as pluripotent stem cells. So there are different types of cells, stem cells, and a lot of them that most people have probably heard of are what are called multipotent stem cells. These are stem cells that are found in the body. A lot of your organs will have these stem cells, and they can only become a few types of cells in the body. Like there's like muscle stem cells, so they become muscle. There's neuronal stem cells that become different types of neurons. So we work with the cells called a pluripotent stem cell, which can become any cell type found in the body, which is good because that gives it a lot of power. But it also means that we have to be a lot careful. There's a lot more ways for the process to go wrong, if you will, because there's now, instead of there being 3 options for what that cell can become, like in the case for multipotent stem cell, we have over 100 different cell types in the body. So we need to make sure that we're, not doing the 99 cells that we don't care about and just doing the one cell that we care about, which is the insulin producing beta cell. And so really the main application that we care about in my lab with the stem cell research is twofold. The first is that we're really interested in using these lab grown insulin secreting cells as a replacement of the cells that are either not keeping up with the workload in the case of type 2 diabetes or are dead in the case of type 1 diabetes. Basically, we want to replace the patient's own cells with insulin secreting cells that we have grown in the laboratory. There's a lot of where the engineering comes into play, because we do these cultures and these bioreactors that can make billions of cells at a time, which you would need in order to give enough cells in order to treat a person with diabetes. The second thing, which kind of touches on the discussion we were having a few minutes ago, is that we really think this is a good system to allow for us to study the initiating events of diabetes. Like, why does the immune system decide to attack the beta cell? Or why do beta cells, or why are they not able to keep up with the needs, the insulin needs of people with type 2 diabetes? And since we're producing these cells in the laboratory, and these are human cells, we're able to directly study and interrogate them with a wide range of tools and technologies in order to really get a brand new understanding of what causes diabetes, with eventually the hope being that we could prevent diabetes from ever forming in the first place.
That is fantastic. Thank you, Jeff. And I want to thank Dr. Paul Durham, who submitted that question. He's a great researcher down at Missouri State University, so I'm glad he's joining us today. So Leslie, you are the managing director for the Advanced Health Data Research Institute at St. Louis University, which is a comprehensive center for data-driven innovation and research aimed at improving the health of individuals and populations. So tell us about what the Institute is doing in the diabetes field.
Yeah, so I'm going to start a little bit about what the AHEAD Institute is, kind of our foundational principles, because they're not entirely diabetes specific, but we have a lot of diabetes work going on. And really, our institute was founded by the idea that there is an immense amount of data that is collected in the real world, in real world processes. You go to your physician, there's a clinical encounter that takes place. All the information that happens in that clinical encounter is then put into a computer for both documentation purposes and billing purposes, right? But they also have the ability to then say, what can we learn from that information about how patient care is delivered? And what can we learn about outcomes of particular treatments as they exist in the real world? So this is kind of the complete opposite side of what Dr. Millman was talking about, because now we're talking about for patients who do have diabetes, what's happening with their care in the real world. How are they interacting with primary care providers? How are they interacting with specialty care providers? And what types of treatments are they being given? And how are they responding to those treatments? It also allows us to look at differences in subpopulations. So if we have patients that are low socioeconomic status and on Medicaid, are they receiving the same types of health services as people that are on a commercial insurance plan? And so it's the work that's going on in the AHEAD Institute right now is less on the prevention of diabetes and more on how do we ensure that patients that have diabetes are receiving the best care that they can get. How do we look at disparities and inequities across different populations in care? And then how do we use that information to say what changes can we make to how we interact with these patients, or what interventions can we do in order to ensure that we have equitable access to care, that patients are getting the care that they need when they need it, and that we can also look at, are those treatments that we're offering, are they working the same way in the real world as we thought they were when these randomized controlled trials were done? So just a particular example of that, when insulin pumps. So when insulin pumps were going through the randomized control trials, the populations in those trials were primarily white and fairly affluent overall. So do the results that translate from those trials translate to a population like we see at SLU, who may be at lower socioeconomic status, primarily non-white populations. And so there's a lot of questions we can ask using this real world data about how what we know from a trial translates to what's happening in the real world. And a lot of times we would love to say that it perfectly translates, but it may not. So some of the research going on at our institute was looking at whether or not lower socioeconomic patients were actually receiving access to insulin pumps the same rate at people who were on commercial insurance plans. So we were looking at Medicaid patients compared to those who were on commercial insurance. And it turns out the Medicaid patients, despite the fact that these pumps are being covered by Medicaid, are much less likely to actually receive a pump. We don't know if that's because of a prescribing bias by physicians or if it's because of a patient preference to not want it. That's a lot of research to go on in the future, but we do know there's a difference in who's getting access to this type of diabetes technology. And that has been posed as one reason why we see differences in long-term outcomes and how successful these patients are at managing their blood glucose over time. And so we want to keep investigating that to understand why we're seeing these differences, as well as are those differences why we're seeing larger differences in the population level as to who has better control long-term.
That is such important work. Thank you, Leslie. That's great information. I want to remind everyone, if you're just joining us, to please put any questions that you may have in the chat box, and we will try to get to as many questions as we can at the end of the discussion. So Derek, as the former president of the National JDRF, can you tell us about some of the other research projects taking place around the country?
Well, I could spend hours on that topic happily. So I'll just mention a couple of things and do a little teasing, and then we can see where we want to go from there. But first, I'll mention that that JDRF last year funded about 75 different clinical studies, or parts of clinical trials. In other words, tests of some therapy or another in patients, which is important because that means that that therapy is reaching a place where it is actually potentially on track toward ultimately being in people with diabetes commercially and broadly. And so when I look at the work that JDRF, but in general in this field, we think about three things, and it's not unique to type 1 diabetes or diabetes in general, certainly. One is curing the disease, another is preventing the disease, and the third is helping people do better with the disease until the day comes that they are cured. So let me talk about each of those just very briefly. With Jeff, you've already heard an example of work that's happening toward a cure. And look, I don't want to get into too much around semantics about what constitutes a cure. It means different things for different people. For some people, if they had to take drugs, a pill every day or something like that, but that magically made their blood sugar be normal, that might be a cure. For other people, they want to be able to walk out of a clinic, never have to do anything again, or at least for some meaningful period of time, and have their blood sugar and be fine and have them not worry about complications, for that to be in their minds an honest to goodness cure. But regardless, the point is there are therapies that are being tested right now that are going to help the person lead a far more normal life, and in fact, in some ways may well be considered to be a cure if they happen. So when I talk about a prevention versus a cure, they're actually very closely related biologically, because the immune system has a lot to do with this disease. And so if we can find ways to have the immune system get its act together and not continue to destroy those cells that Jeff was describing, those beta cells, then yes, that's a way to prevent this disease for someone who doesn't already have it, but it's also a key step toward a cure. For a cure, we need to have two things happen, really. We need to stop the destruction of those insulin-producing cells, and then we need to make sure that the person has enough of those cells. Because by definition, by the time the person becomes insulin dependent, too many of those cells have already been wiped out in that person's body. So Jeff, in his work, is helping us figure out the second step of that, which is to make sure a person has enough of those cells. And then so if we can marry that with an approach toward modulating the immune system so that the immune system stops being tricked into thinking that it should attack and destroy those beta cells, those insulin producing cells, then we have the two pieces that we need for a cure. I'm excited about the fact, therefore, that when I look at in the area of prevention and immunology, there are a number of different therapies now that have been shown in different clinical studies that they will at least significantly delay the onset of type 1 diabetes and may, in fact, over time prove that they can prevent the disease altogether. For example, teplizumab, I always have trouble with that, teplizumab, is at the FDA right now, and the hope is that that will be commercialized in the third quarter of this year. It's from a company, Prevention. It has been shown to delay the onset of type 1 diabetes by several years. In fact, there's probably at least a half a dozen therapies that have been shown in clinical studies to delay the onset of type 1 diabetes by multiple years. Jeff mentioned earlier about beta cell health. And so separate from the immune system, we think, would be an approach that leads the beta cell to stop being attacked or asking the body to come and attack it, because strangely, there's actually this signaling process that occurs where the beta cell, in a way, kind of says to the body, come and get me. It sends a signal out in the form of a protein that triggers this cascade of events that ultimately leads to the beta cells being attacked and destroyed by the immune system. So, for example, there is a drug that's been on the market for 50 or something years, which is actually a high blood pressure medicine, verapamil, which has been shown that it seems to disrupt that signaling mechanism from the beta cells out to the body to say, come and attack me. So exciting progress in both of those areas. But meanwhile, we recognize that even if the magic elixir is sitting in a test tube right now, it's still many years before it's going to be commercial that would be the honest to goodness cure. So we need to help people lead fuller, safer, happier, higher quality lives until that day comes. And that's obviously what Leslie's focusing on a bunch when she's looking at the different tools and making sure not only that they are effective and approved by the FDA or other regulators around the world, but also that then they're prescribed, they're used effectively, and they have the impact that is desired. And Leslie's exactly right that we see a significant difference in terms of the way things go, the different outcomes for people. And even though we see better devices and fancy systems now that are really remarkable in their integration that will allow a person to have much tighter control and a lot less hassle than used to be the case, sadly, if we use the metric of A1C, hemoglobin B A1C, the average for a person with type 1 diabetes is essentially now the same in this country as was the case 10 or 20 years ago, which is a head scratcher. How can that be? And it's because, as Leslie's trying to answer these questions, we still need to figure out how to get more of these systems in the hands of people that can use them and then help them use them more and more effectively. And there's a lot to unpack in that, and I'm happy to go into that more, and I'm sure Leslie is too. So anyway, I could go on and on, but the point is you got cure, treat, and prevent. In all three of these areas, there are significant steps that are being taken and a lot of progress.
No, I love it. Thank you, Derek.
If I could ask Derek a follow-up question, actually, I had never heard about that A1C not having actually been improved over the last decade. That's pretty almost, I must say, crazy, considering the advances that have gone on with device technology. So I'm curious to know, besides fancy gizmos, what has actually improved in the treatment of type 1 diabetes in the last decade? Have these technologies maybe made people made their treatment and management of the diabetes less of a burden, perhaps? Or how would you think about that?
So think about a bell curve with the x-axis being the A1Cs for a lot of different people, and the frequency, and the y-axis is the actual A1C number. And what has happened over time is we see a wider spread, a greater standard deviation among the population. In other words, there are people who are the intensive users of technology, the tools. They're definitely doing better. Their A1Cs have come down and life has gotten easier for them. My wife Emily and I I don't know how many hundreds and hundreds of times we set our alarms in the middle of the night to wake up and go test Turner, because there wasn't a continuous glucose monitor that we could just check by our beds that was signaling back to us from Turner. And there wasn't this integration of a system that would automatically bolus Turner, give him insulin when his body was having high blood sugar, or shut off the insulin administration when his body had low blood sugar. So we were guessing and testing and all kinds of things all the time. So for that population segment that is really at it, life is a lot easier, and the numbers have in fact gotten better. But it's for so many others where the treatment gets harder, and unfortunately, partly about our medical system, it's difficult to get good care. And now I should definitely turn it over to Leslie, because this is
I was going to say that this really illustrates how important Leslie research is. So I'm really glad we're talking about this.
Exactly.
Well, I appreciate that. I do. There's surprisingly low uptake of diabetes technology. And from the work that we've been doing, I don't have a reason as to why. What we do seem to know is that patients who are seen by endocrinologists are more likely to have access to diabetes technology. We also know that there is a significant lack of endocrinologists across the United States. So there are many patients who actually don't have access to one, or are just seen by their family practice doctor or their primary care doctor. There's nothing wrong with that particular approach. I am in no way, but I'm saying that endocrinologists are usually more up to date on what diabetes technology is there, and are more likely to prescribe it. There's definite learning curves using these technologies. And so I do think that in some instances, there are somewhat artificial barriers to access to this technology, because they do require so much, they require a significant amount of training at the beginning to use. And so and what we don't know, there's a possibility that some patients are just unwilling to undergo that. There's also a possibility, and this is brought up in the literature a lot, but not very well investigated, that there is a provider bias as to who they offer these services to. So there may be situations in which providers are saying, I don't believe this patient is capable of handling it. So, and we don't want to deal with pump failure, so we're just not going to offer it, and we're going to keep them on their multiple daily injections, right? All of that still needs to be teased out and understood. And a lot of that has to do with human behavior and ways in which implicit bias interacts both from a patient side and how they interact with their providers and telling them what they think is good, or asking questions, or availability or willingness to ask questions, as well as provider biases to what do I think this patient is and is not capable of.
Right.
And then you add on top of that the training that it takes, and do I require someone before I'll give them a pump to come in and do a half day or a full day of diabetes training education on that device before I'll give it to them? Have I considered the fact that they may not be able to get the transportation to me in order to be able to do that? So then how have I excluded that person from having access to this device just because I insisted they come here instead of me having a diabetes education program that will go to them? So as far as understanding the kind of distribution, uptake on these devices is fairly low across the board. But then when you look at it by socioeconomic level, by insurance status, and by racial distribution, then you see some significant gaps in who is and who is not gaining access to these technologies, and how well that technology may or may not work within that population.
Exactly. I'll say two quick things following up on that, agreeing completely with everything you just said. Two interesting facts, I think. One, and they're related. You mentioned the shortage of endocrinologists, who, for those who don't know, that's the specialty that particularly focuses on diabetes. Endocrinology is the second least lucrative specialty in medicine. And related to that, diabetes clinics are money losers all around the country. And there's a very good reason for that. The average visit, supposedly, according to a Society of Endocrinologists, should be about 40 minutes with a patient in order to go through the data and think about better outcomes and stuff. If that visit goes longer than 11 minutes, it's a money loser for that clinic, unless there are lots of procedures done. And so therefore, when they're just having this quality conversation between a doctor and a person with diabetes, which is what that person needs in a lot of cases, the clinic is losing money. And so this becomes a very difficult area systemically to receive the support that it needs to receive. There's some good questions I'm seeing online. Is there a test that can indicate whether a person is at risk for type 1? There is. In fact, I can tell a story about our other son. So I've mentioned Turner, our son who has type 1 diabetes and has for 16 years now. Our other son, William, through a screen through a program called TrialNet. It was determined, gosh, 13 years ago, I think now, that he had multiple autoantibodies. So that's a biomarker that is used to determine whether a person is particularly at risk for type 1 diabetes. And at this point, there are 5, at least I believe that's still the number, 5 autoantibodies that are screened. Back when William was screened, there were only 4. William had a few of them, and that meant he was significantly at risk. In fact, JDRF has now started a program where anybody can participate. And if you go on the JDRF website, jdrf.org, you can see a way that you can then participate and get the screening kit that you need in order to determine whether you or a loved one is at risk. And that's really helpful information, both because because this is so familial in a lot of cases. In our case, we have 7 people in our family with type 1 diabetes. And so obviously there's a genetic thing going on. And I mentioned all the other autoimmune diseases. Also, it allows you to see whether a person perhaps would make sense to go into one of these clinical trials that I mentioned before, where prevention can be so great. There's also a comment here about low carb diet, and Leslie maybe is going to have some comments on that too. But it's, you know, different diets are going to be right for different people. But Darnell, you're exactly right. I talked to a number of people who can make it work with a very low carb diet, who have extremely impressive results with their A1Cs and with their time in range and all. So you're right. Leslie, you want to add to that?
Yeah, I think there's kind of 2 points. I've spent a lot of time talking about our work with insulin just because that's what's kind of the most pressing and ongoing at this point in time, as far as the work that I've been doing. And I do want to point out my colleague, Alexis McKee, who has been, she is an endocrinologist who's been instrumental in all this work. And I don't want to not give her credit for that while we're here. But as far as the diet goes, it has some really interesting influences on how we can look at real world data and use real world data to understand what's happening in patient care and treatment, which is going to only partially address what you're talking about. Diet is exceptionally important. When we're looking at how things are in medical records and in kind of claims data, so that data that it happens when you pay for something through your insurance, we don't really have access to information unless you went and saw a dietitian or a nutritionist. We can see that that was billed for, but that type of information doesn't explicitly exist in the type of data the Head Institute uses. So a lot of our focus really is on technology and insulin treatments and or oral antihyperglycemics or things that we can measure in our data. And so that's when that's why a lot of the research out of the Head Institute is not addressing diet, because it's hard to, you can't really measure it in the types of data sources that we have. That being said, I think the point is very well taken, especially if you try to track time and range, which is really becoming the bigger metric of how we can establish whether or not someone's well controlled. HbA1c has been used for a long time just because it's routinely collected. You're supposed to get it done every 3 months. We can look at it. We know it's an average of your last 3 months blood glucose, but it doesn't tell you if you're spiking up and down, right? Exactly. And we know that spiking up and down has impacts on how you do long term. And so with the advent of continuous glucose monitoring and that data, once people actually start to use CGMs more regularly, as that data becomes more available, we'll have a lot more information that researchers can use to track, okay, if someone's has high fluctuations versus someone who has low fluctuations, even if they have the same HbA1c every 3 months, are we seeing better or worse outcomes in someone who maintains that kind of tight control around their ideal blood sugar, or are we looking at, which my guess would be yes, but we'll have a lot more evidence for looking at the impact of diet and or when you have a meal as we start to get more of that data in and we can analyze it.
Yes. Agreed.
Well, I was going to ask Jeff to address the next question. We're getting great questions. Thank you. And talk about are people with diabetes more likely to have more serious risks from COVID-19? Is there a correlation there?
Yeah, there's definitely been, this is not an area that I study, but I've been following it somewhat to a great extent. And there definitely has been correlations with outcome of COVID-19 and the patient being diabetic in terms of like there being worse outcomes if you have diabetes. I think there's a lot of controversy. I don't know if the other panelists have more recent papers they can cite about this, but my understanding is kind of a lot of controversy in terms of like what is actually causing that. I know one of the scientific questions that seems to keep on seesawing back and forth is this idea: does the COVID-19 virus actually, the coronavirus, actually infect the islets or infect the pancreas or affect the beta cells? And there's like a pretty heated like feud going on in the literature about that, where people say no, that doesn't happen at all, and people say yes, it does happen, and it really hurts the beta cells. So it's one of these like scientific. So there's a lot of scientific murkiness right now in terms of the literature. You probably find papers support whatever scientific hypothesis you may have on the topic, but it does point to the overall issues here of needing to make sure that we have populations of people and populations of people with diabetes who are as healthy as possible, because we could have things like a pandemic come that might put them at more risk on top of the usual risk they have because of their underlying metabolic disorder.
Yeah.
Hey, Jeff, so staying on that, one of the things I've wondered about is the H2 angiotensin being an important receptor in beta cells, as I understand it. And that's been identified as an aspect of COVID-19 too. And I was curious whether that maybe would be a reason why COVID-19 might be tougher on beta cells than on some other cells in the body.
Yeah, there are, again, I never looked at this myself, so I don't have a opinion on it, but there are papers that claim the receptor exists on the beta cells, and there's papers that claim the receptor does not exist.
Oh, okay.
And I thought like half a year ago it was settled. I was just flipping around so much, I'm getting my order mixed up here. It seemed to be pretty much settled about 6 months ago one way, and there was like some recent papers that came out the last month that seems to be again pushing back against that. And that maybe I doubt anybody is doing the experiments wrong, and this actually might be more of a reflection of the heterogeneity that exists among patients who, in the way their islets are, or the various models that people are using for this. It might really just be a reflection of population heterogeneity rather than half the scientific community being wrong on the topic. That typically doesn't happen.
Yeah.
Mm-hmm, definitely.
The type 1 diabetes community is pretty upset right now. I will say that that in certain states, at least, type 2 diabetes is being treated differently in terms of the pecking order for vaccinations versus type 1, because there is certainly no reason to think that that the outcomes would be better for one population than the other, it seems. And so there's been some push that way.
I did actually read a paper yesterday about COVID-19 and type 1 and type 2 diabetes. And from the limited data they have, the type 1 patients are having some pretty severe outcomes.
Yeah.
Yeah.
And then the other question is, will we see a increased onset of type 1 down the road, perhaps because of, for example, the mechanism of the H2 receptor in beta cells being affected by the COVID-19 infection?
Yeah, and there was a paper early on that really, with a very limited patient number, drawing a correlation between increased incidence of autoimmune type 1 diabetes after COVID-19 infection. I don't think that there has been a rigorously done study to show that is the actual case or not. Though there is evidence out there for these other viral infections being associated with causing type 1 diabetes, and some of the inheritable gene mutations that are correlated with type 1 diabetes are actually associated with genes that affect how a cell responds to a viral infection. So there's definitely reason to think that there could be a correlation there, but I don't think the clinical data has shown that. But of course, diabetes takes years to form, and we might not really know the answer to this until 5 years from now.
Right. And with regard to viruses, so Coxsackie virus is a virus that has been shown to be a vector for type 1 diabetes. And in fact, that same company I mentioned before, Provention, also has now a phase 1 study. I think it's just about to start, looking at a Coxsackie vaccine as a means of reducing the incidence of type 1 diabetes.
There's also a question, I know you were talking about the COVID vaccine, and they're asking, in the state of Missouri, are type 1 patients able to get the vaccine no matter what age they are? Are any of you familiar with that, or do you know?
I'm not certain.
I feel like the answer is type 1 is included in it. They don't specify between type 1 or type 2 diabetes, and I am 90% positive that is true, but I would rather look it up than tell you that I'm lying. But I am next to positive Missouri doesn't differentiate between type 1 and type 2.
And then I want to address the other question. My 11-year-old was diagnosed with type 1. In May 2020, we started using Dexcom G6 in December, and aside from taking a screenshot every day of his 24-hour period of results, how else do you suggest we log or track his blood glucose?
So I would suggest that you talk to your doctor about that. So the way that we would use that information from a research perspective is quite different from a way a clinician or a parent would use that information, and I am not at all qualified to discuss how you would use it. We, as a researcher, we can look at those long-term trends and draw larger associations between what's going on. But it sounds to me like you're doing what your physician has recommended, and they will be able to help you understand and interpret that data that's coming out of your Dexcom to make better decisions about what you're doing with your child.
I'll just say, as a parent, and our son was diagnosed when he was 10, a couple months before he turned 11, so close to the same age. It does get easier. I'm sorry for what if your year has been like my year was that first year, and Emily, my wife, I know it's a hard time, and you all feel fragile. And I promise that you do settle into more of a normal. Management of this disease is never easy. My gosh, it's like catching a constantly moving butterfly. But I promise you will settle into more of a normal, most likely anyway. And that's certainly been our experience, and I think the experience for most people. So hang in there.
Thank you, Derek. So I want to ask all of you, what are the unique challenges you see in diabetes research? Is it the funding? And I don't know who wants to go first on that question, but Derek, you want to jump in there?
Can I just say yes on funding?
Yes.
I'm going to push Derek out of the way there for a second. Yes.
Yeah.
No argument there. I'm with you there. I'd say the heterogeneity of the disease is at least the first thing that occurred to me. Even within type 1, I don't think there's just type 1. I think most experts agree there's type 1A and 1B and 1C and whatever. And over time, we're going to characterize that some more. And we see how this disease manifests itself so differently in different people. 2 people will have autoantibodies when they're little kids, and 1 person develops type 1 or becomes insulin dependent, you know, at age 2, and another one doesn't until age 50 or 60. In my own family, we have people who are diagnosed as young as in their diapers and as old as 51, I think. So I think the heterogeneity is really a big challenge in this, because you're talking about a lot of different issues you're dealing with among all these people. And then tied to that, I'll just cheat and have a second one, which is you're talking about real world, real life experiences. And so that makes the data so noisy, which is kind of what Leslie's driving at too. So even if you've got the right tools and understanding or whatever else, it's so darn hard still.
Mm-hmm. And Jeff, what would you say to that as far as challenges?
I could speak hours and hours about the funding challenges in particular, but maybe to speak about that a little bit of a broader context. When you conduct research and you have like a research lab, and it doesn't matter if you're focusing on clinical care, more like Leslie's doing, or doing more like wet lab research, is what my lab's focuses. You have a reality that you have to work within, that you have to raise funds. You have to come up with ideas. You have to raise funds to support the research. That funding has a period of time. Typically it's I'd say on average about 3 years, but never goes any longer than 5 years. And so you're kind of constantly in this cycle of needing to, like a scientist. Like I lead a research lab of 12 people, but most people who are not scientists are surprised to learn I don't do research personally. Like pretty much all my time is spent in terms of reporting results, which is obviously important, but also on getting the money to sustain the laboratory. And most of that comes from the government, and the NIH is definitely, we're really lucky in the United States to have a robust scientific funding agency like the NIH that funds biomedical research. But even then, oftentimes the types of funding they want to do to projects they want to fund are very narrowly focused and on very like detailed studies of small mechanisms, which is important to do, but it makes it very difficult if you're trying to do more translational research, which is really where the focus of my laboratory is, to kind of go beyond like understanding the details of a particular pathway, which may be important, to go into putting together multiple different pieces of evidence into a process for producing beta cells, like in my case. We're also very fortunate to have agencies like the JDRF that really does a good job, I would say, in kind of bridging that gap in terms of the what the government's not willing to do, and JDRF is willing to fund the more translational sort of work. But it makes it kind of a unique thing if you're really focused on research that is trying to be for going beyond basic scientific questions to something that's more closer to the patient or more translational. It can be really hard to get the supports to actually do that as much as you may want to run a lab.
Mm-hmm. And I have to ask Leslie, like, what are the challenges that you have, some of the roadblocks in assisting people in the community who have diabetes? I mean, I'm sure some people struggle with the cost of insulin and so forth.
So the cost of insulin is definitely one. I was talking with Dr. McKee this morning specifically about that, and she was going on a little tirade about the Medicare donut hole and what that does to her older patients and their ability to access insulin. I think one of the other really big challenges, both to the research as well as to the patient care side, is the fact that we're talking about a disease that requires a lot of behavioral change, right? And it's not cut and dry, and it's not, I can just take a pill. And so you are now asking patients to come into a clinic to provide them with information on how to manage their disease. And then you're asking them to go back into their real life and to be able to make all of those changes, oftentimes without support systems that they need. Or for those of you who are familiar with the idea of social determinants of health, it's the general gist that your lived environment, all of the things that socially interact with you, influence your health, not just in your ability to take care of yourself, but also can have direct impact on how healthy you are. And so either both from a research as well as from kind of how research intersects with this clinical practice, a lot of the barriers are just those barriers that exist with being human, living in environments that may not be conducive to the changes that you need to make, either to prevent progression of disease or to make sure that you can maintain your health status. If you have type 1 diabetes and don't have the ability to get your insulin, you are going to end up hospitalized. That is going to cost you and the health system quite a bit of money, right? And those are the types of kind of policy factors that we also need to be considering when we're talking about how patients are able to care for themselves and how they're able to exist in their own environments and keep themselves healthy, knowing that they live in situations that may not be conducive to doing so. And that's really, I think, one of the barriers, not just insulin access. The barrier is the entire scope of social determinants, and as well as it's really hard to change your diet. It's really hard. We all know that, right? And we ask people to do that in order to stay healthy, and then don't provide them with the supports, or they don't necessarily have access to the supports they need to be able to do that. And so how do we, as researchers that are working within systems and trying to find better ways to reach patient populations, as well as make sure that our clinicians are providing the care that they want to be providing their patients, how do we work with all of those different factors to ensure that everyone's getting what they need to stay healthy? And it's a really complex system.
Yeah. And there's the realities that it's just plain expensive to eat healthy, to that particular thing, for example.
Yeah.
I know that, yeah, the real world aspects of this are so hard.
I want to make sure I'm respectful of everyone's time. And my last question, and it kind of ties in with a question we had over here, is how patient advocates can get involved with moving policy forward, therapies forward for diabetes. As someone said in the chat box, who's advocating for diabetes research, access to care, lower cost for insulin with Missouri policymakers? So what advice would each of you have for patient advocates, people who care about seeing this moved forward? Derek?
Sure, I'm happy to. But so JDRF is obviously a leading advocacy group, but that's at the federal level, not at the state level. And so JDRF focuses a lot on issues such as availability of care and certainly funding of type 1 diabetes research. At the state level, it's a lot harder, frankly. I think there's a fair point to be made that there could be some more organization around this community being more active in the state of Missouri, because, as I think probably many people are aware, something like Medicaid is a state program. It's not administered by CMS nationally. There are 50 programs around the country. And when we talk about a lot of insurance aspects, that again can end up being variable by state. So, you know, I don't have a great answer for you other than to say it's important, and I think we need to do better.
Yeah, and I would encourage people to get involved with Missouri Cures. I mean,
I was gonna say, isn't the answer Missouri Cures?
I mean, that is our main focus. That's why we exist. We love doing these educational seminars, but we exist to protect all medical research in the state. And when issues come up, we try as much as we can to get involved with those. We keep updates in our newsletter. We send out policy alerts and so forth. So if you're not on our database, please go to our website, missouricures.org. Sign up, it's free. And we need your help. We need people who care and advocates, because when bad legislation pops up in this state, the only thing that we need your voice to help shut down all of the bad stuff, so to speak. But I just want to say thank you to Jeff, Leslie, and Derek for your passion, for caring so deeply about this issue, and for your insight and your advice today. And we will do a follow-up. This is being recorded, so we will send this out. It will be on our website. It will be on our YouTube channel. Anyone who signed up for this seminar, sometimes things pop up and they can't participate, but we do a follow-up email with the recording. We will include a bunch of the questions that were in the chat box with information. And so I really appreciate everyone being with us today. So thank you so much.
Thank you, Dena.
Thank you.
Yeah, thank you, Dena. Great job keeping us under control.
Yes, it was lovely to be here.
Thank you. Mhm.